Welcome to the Muscle Growth Podcast. Hypertrophy, strength and performance, health and longevity. Bigger, stronger, better. The Muscle Growth Podcast.
Hi, everyone, and welcome to the Muscle Growth Podcast short episode edition, where today we will be reviewing a scientific paper called Human Skeletal Muscle Possesses an Epigenetic Memory of Hypertrophy. My name is Roscoe, and I will not be your host today. However, I will be your curator, and I really hope that you guys enjoyed this episode. Cheers.
So we've all heard the phrase muscle memory. You take a long break from the gym, you come back, and the gains seem to come back faster.
Right.
But the question we're tackling today is, what is that memory? Is it just your brain remembering the motion, or is something deeper, something molecular happening inside the muscle itself?
That is the exact question. And we're not talking about, edge cases like steroid use or development from birth. We're asking about regular adult humans.
Okay.
Does adult human skeletal muscle have an epigenetic memory of training?
Right. Epigenetics, so not the DNA sequence itself, but the little chemical tags that sit on the DNA and tell the genes whether to switch on or off.
Exactly. And the study we're looking at was designed specifically to test this. It's a really clever model.
So what did they do?
They took a group of untrained men through 3 distinct phases. First, a 7-week training period they called loading.
Okay. Building the muscle up.
Then a 7-week break. Total inactivity. They called that unloading.
Letting it go back to baseline.
And finally, the test. Another 7 weeks of training, which they called reloading. That's where you'd expect to see the memory effect, if it exists.
The ultimate test of retention. Did the muscle forget the lesson once the physical gains disappeared?
Let's find out.
All right, let's start with what you can actually see and measure. The physical muscle gains. We've got 8 healthy guys, all previously untrained, going through this whole 21-week cycle.
Right, and they're taking muscle biopsies at every key point.
So phase 1, the initial 7 weeks of loading, what kind of growth did they see?
They saw a pretty significant 6.5% increase in lean lower limb mass. That's a solid, a very respectable result for 7 weeks in beginners.
Okay, standard stuff. Then comes the break, 7 weeks of nothing.
And this is crucial. During that unloading phase, their lean mass dropped by 4.6% from that peak.
So they lost most of what they'd gained.
Pretty much. The sources are clear that they returned significantly toward their original baseline. The physical gains were, in a sense, erased.
Which you need.
Yeah.
Right. Otherwise, you can't prove it's a molecular memory.
Exactly. So now, the big question. Phase 3, reloading. They get back under the bar. What happens?
This has to be where the magic is.
And it was. The reloading phase produced by far the largest muscle gain. Compared to their original starting point, they saw a massive 12.4% increase in lean mass.
Wow. But was the rate of gain faster? That's the real proof of memory.
It was, and they measured it directly. When you compared the rate of growth in the first phase to the rate in the third, the reloading phase resulted in a significant 5.9% greater increase.
Okay, so that's the physical proof right there. The muscle didn't just regain mass, it did it with like a hypersensitivity.
That 5.9% difference is the number. It proves something was retained molecularly.
All right, so we've got the physical evidence. Now we have to find the molecular ghost in the machine. What is the mechanism?
We're looking at DNA methylation, and just as a quick primer for everyone, you can think of these as little dimmer switches on your genes.
Okay.
High methylation generally means the gene is dimmed or turned off. Low methylation or hypomethylation means the switch is on, the gene is active.
So for muscle growth, we want to see lots of hypomethylation on the right genes.
Precisely. And after that first loading phase, the muscle responded. They found 9,153 sites across the genome that became hypomethylated.
A clear signal. The body is turning on its growth machinery.
But here's where it gets really weird. During the unloading phase, when the muscle was shrinking back down, the genome didn't revert. It kept a very similar number of sites, about 8,900 in that hypomethylated on state.
Wait, that's completely counterintuitive. The muscle is getting smaller, but the genetic instructions for growth are staying active.
That's the memory. The body is choosing to keep that instruction set on standby even when it's not being used. It's inefficient in the short term, but brilliant for the long term.
So what happens during reloading?
An explosion. This is where the memory pays off. In the phase where they saw the fastest physical growth, the number of hypomethylated sites more than doubled. A staggering 18,816 sites.
Twice as many genes being activated for growth precisely when the muscle was growing fastest. It's like the first training period was a primer, and the second one just set the whole system off.
And you can see it in specific growth pathways. The PI3KKT pathway, which is, a master regulator of protein synthesis,
Right.
it had almost twice as many activated sites during reloading compared to the first time. The machinery wasn't just on, it was supercharged.
Okay, so the whole genome is primed. But let's get specific. There must be certain genes that are the true keepers of this memory, the ones that stayed on during that 7-week break.
There are. The researchers identified a group they called cluster B, and you can think of these as the stable blueprint genes.
The ones that hold the memory.
Exactly. The key players they identified were genes called AXIN1, GURI2, CAMK4, and TREF1.
And their pattern was different.
It was definitive. They became hypomethylated, turned on after the first loading phase.
Okay.
They then maintained that low methylation status through all 7 weeks of the unloading break.
So they stayed on, even when the muscle was shrinking.
Right, the light was kept on. And then when reloading started, their gene expression just shot through the roof. It was the largest, most cumulative increase of all.
So what do these blueprint genes actually do? Let's take one. What about AXIN1?
So AKN1 is involved in managing cell structure and differentiation. It essentially helps the muscle cell know how and when to rebuild itself properly. Keeping it on standby means the muscle retains the capacity for rapid, organized reconstruction.
It's like keeping the architectural plans open on the table, just in case.
Perfect analogy. And then you have TRAF1, which is maybe the most direct link to your experience in the gym.
How so?
TRAF1 is involved in inflammatory pathways that get activated by damaging exercise.
So muscle soreness.
Basically, yes, the physical stress of lifting. What this shows is that the first exposure to that stress leaves a lasting epigenetic mark. The gene that responds to exercise trauma stays primed, ready to react much faster the next time.
Okay, this is where it gets a little more complicated because not all memory works that way. There's another group of genes, cluster A, that behave differently.
Right. If cluster B is the stable blueprint, you can think of cluster A as the hypersensitive alarm system.
Hypersensitive alarm. I like that. So they didn't stay on during the break.
No, they didn't. During unloading, they went quiet. They reverted back to baseline levels of methylation and expression. The alarm turned off. But when the reloading stimulus hit, they screamed louder than any other genes. They showed the largest, most dramatic increase in both hypomethylation and expression.
So the first training period didn't keep them on, it just made them sensitive.
It primed them for an amplified response. The key genes here that correlated with the actual hypertrophy were UBR5, SETD3, and RPL35A.
Let's focus on UBR5 because this one is a real head-scratcher.
Yeah.
UBR5 is an E3 ubiquitin ligase,
It is.
which, for anyone who knows a little muscle biology, that's a huge red flag. Those are the proteins that tag other proteins for destruction. They cause muscle atrophy.
Exactly. They're the demolition crew. You expect to see UBR5 activity go up when muscle is breaking down, not when it's building up.
So seeing it spike during the biggest growth phase is completely backwards.
It's totally counterintuitive. And yet, UBR5 had the single most distinctive relationship in the whole study. During reloading, it had the largest drop in DNA methylation and the largest increase in expression. It was the most on gene.
The correlation with the physical gains must have been off the charts to make sense of that contradiction.
It was stunning. UBR5 alone accounted for 33.64% of the variability in muscle mass changes.
A third of the difference between individuals was explained by this one supposedly destructive gene.
Yes. It strongly suggests that UBR5 has a completely novel, undiscovered role in promoting muscle growth in adults. It seems the body has repurposed a demolition tool into a key part of the construction crew.
That raises a question about speed. We're talking about 7-week blocks, but how fast do these changes happen? Could, a single workout do anything?
This might be the most practical and exciting part of the study. They tested that exact question. They took a biopsy just 30 minutes after one single heavy workout.
Yeah.
Just that one session was enough to cause a huge shift. They saw over 10,000 sites become hypomethylated almost immediately.
Instantly. Your DNA is responding that fast. Were any of our memory genes in that group?
They were. Key genes, including GRAK2 and that inflammation-related gene, TRAF1, were flipped to a hypomethylated state within 30 minutes.
Okay, here's the billion-dollar question. Did that change at 30 minutes actually do anything? Did the muscle start building more protein right away?
No, and that is the critical point. There was no change in gene expression at the 30-minute mark. The switch was flipped, but the machine hadn't started running yet.
But did the switch stay flipped?
It did. That initial change, that epigenetic mark from one workout, was maintained for the entire 22-week duration of the study.
You're telling me that one single training session can set a chemical marker on your DNA that lasts for almost 5 months?
That's what the data says. It's an early biomarker, a molecular signature of future potential, just sitting there, dormant, waiting for you to come back and train again to really activate it.
So bringing it all together, this really confirms it. Human muscle has a true molecular memory.
It absolutely does. It uses these epigenetic changes, specifically this hypomethylation, to remember both the acute stress of one workout and the chronic stimulus of a training program. And that memory makes it respond so much better next time.
And it's really two types of memory working together. You have the stable blueprint genes, like AXIN1 and TRAF1, that just stay on, keeping the plans ready. And then you have the hypersensitive alarm genes, like that weird one, UBR5, which reset but then explode with activity when the stimulus returns.
It's a two-pronged strategy. Your muscle fibers aren't just adapting physically. They are literally rewriting their own operating instructions to be faster and more efficient in the future.
Okay, a final provocative thought for you to take with you. If a single resistance training session is enough to set a lasting 22-week epigenetic marker for future growth, what does that mean for you when you're starting a program again after a long layoff? Is that initial deep soreness you feel? Is that just the sound of your genome being chemically primed for rapid future success? Thank you for diving deep with us. We'll see you next time.
Thank you for listening to the Muscle Growth Podcast.